Number 1. Stop the bouncing ball. All right, so a couple of announcements. Number 1, if you haven't discovered already, you will discover when you look on the web page that the camera was mispointed. And I was disappointed because it was mispointed, so that was unfortunate. Hopefully you can get out of the audio what I had to say, but you're not going to see a lot of things on the screen, unfortunately. That was for the lecture on Wednesday. So just pay to come to class. And that's number 1. And number 2, I apologize. I have not been very available to you. I, for the past 3 days, have been interviewing students to be ambassadors of undergraduate research, and I've been in my other office. And so if you come looking for me, you haven't been able to find me. So I decided what I would do is I will extend the period of time that you can request regrades on your exam if you have that to do. So I'm going to extend that through Tuesday. Okay? That'll give you more chance. I will be in my office on Monday and Tuesday, so in my main office on Monday and Tuesday. So I apologize for that. And when I have those situations, I will try to let you know. I just got wrapped up in this, and I didn't let you know about that. So that's my intention. Okay? Okay. I think that's it. So I want to move forward talking about DNA, and we'll talk about RNA today, and we may talk a little bit about DNA replication. So a lot of excitement before the weekend. I hear you. Okay, so last time I talked about the different forms of DNA, and I said we had right-handed form, we had left-handed form, and it really had to do with how the strands interwound with each other, and they are different ways of winding each other. Okay. Now, for our purposes, it's mostly sort of an artifactual thing. Well, if it's in the right-handed form or it's the left-handed form, it's just one more thing Kevin has given us to memorize. But they really do have implications as far as the structures of these things go in the cell. It's beyond what we'll talk about here, but I don't bring it up as an exercise in something. I don't bring it up if it's not important. Let's put it that way. Another thing I want to bring up that's important, and it's kind of hard to see in this figure, is the phenomenon known as supercoiling. So when we talk about DNA structure, we have to think a little bit about the way in which it can appear, the way in which it can exist. And one of the ways in which DNA can exist is in a form that we call supercoiled. So I need to tell you a little bit about what that means. Okay? I've got 2 strands, and they're wound around each other. And you can see the form that we kind of think about DNA existing in. They rotate, they go all the way around. The B form of DNA that Watson and Crick and Franklin, I'll put her name on there, described, has 10.5 base pairs per rotation. Okay? So one rotation of the helix is 10.5 base pairs, meaning that if you went 2 rotations, you would go 21 base pairs. That's what it amounts to. And that form of the DNA we refer to as relaxed. It's kind of like you are hopefully right now. Haven't had an exam yet. I mean, you had one exam. The other one's not far enough away that you're uptight about it yet. So you're kind of relaxed. And it's Friday afternoon. That also helps, right? Okay? If I were to take one of those strands and I were to wrap it or unwrap it around the other, as I could make it more tight or more loose, all right, I would change the number of rotations of the helix. Right? So if I took one strand, brought it back around, then I tied it to the first strand. So I took this guy here, right, took one strand, wrapped it around, and then tied it back. This circle that you see here wouldn't have the same number of rotations that it had before. I would have taken one out or put one in. Well, those are the number. If you remember what I said, a relaxed form of DNA had 10.5 base pairs per rotation. I've got the same number of base pairs, but if I change the number of rotations, now I'm going to change base pairs per rotation. Right? That makes sense? So I've changed rotations. I have the same number of base pairs, so number of base pairs per rotation is going to change. I'm not going to have 10.5 after I do that. Okay? Well, why do I tell you that? Well, what I've just done is I've just strained the DNA double helix. I put some stress on it. And it can go either way. If I put an extra turn in, or I take an extra turn out. Either way, I've strained it. Anytime I've gotten away from 10.5 base pairs per turn, I strain the DNA. All right? Now, the analogy I like to give to that is, imagine you have a rubber band, okay? And you grab one side of the rubber band and you pinch it. And you take the other side of the rubber band and you start putting in some twists. What's going to happen to that rubber band? It's going to get strained, right? And it's going to get all kinked up, right? That's exactly what DNA does. Now, this shows one kink. In fact, DNA may get many kinks. And those many kinks are what we call supercoiling. The stress on the DNA is causing the overall duplex to do something to try to relieve that stress. And the something it's doing is it's coiling the whole molecule up in a different form. So instead of laying flat like this, it's going to start twisting on itself like you see here. Okay? That's what supercoiling is about. Now, that supercoiling turns out to be really important for a couple of reasons. One of the reasons is when this guy gets nice and supercoiled, just like the rubber band starts out laying flat. You start coiling, and what happens? It kinks up. It's going to get very tiny. The size, the overall size changes with supercoiling. It'll get smaller. The other thing that happens, and this actually is a little bit more involved than what I'll tell you why this happens, but I will tell you what happens, is when we put DNA under strain like that, some of the sequences start coming apart. They start coming apart. Now remember, what's the force that's holding the double helix together? Those are hydrogen bonds. And those hydrogen bonds are relatively weak. The weakest links are going to start coming apart. Okay? Now what I didn't tell you last time, I forgot in my lecture, so I'll make sure I mention it here, is that hydrogen bonds do hold the base pairs together. I told you that. I didn't tell you that GC base pairs have 3 hydrogen bonds each, whereas AT base pairs only have 2 hydrogen bonds each. So if we think about the forces that hold together a double helix, all right, GC has a lot more strength holding it together than AT does. And you can start thinking, if this thing is supercoiled, what's going to be the most likely sequences that are going to try to come apart? It's the AT. Okay? And we'll see that AT plays a very important role in the cell when we get to that point. There we go. Okay. Now, another thing we have to think about with respect to the structure of DNA is chromatin. Okay? What is chromatin? Well, I've told you earlier that there's 7 feet of DNA in every cell of your body. 7 feet. Okay? Well, you know you don't have 7 feet out there. The DNA has got to be coiled, it's got to be wrapped, and it gets wrapped around proteins called histones. Histones. The DNA gets wrapped around proteins called histones. DNA is negatively charged. It's full of phosphates. Phosphates at physiological pH are negatively charged. Histones are small proteins that are positively charged. They attract each other nicely. And the histones are shaped so that DNA will actually coil around it, kind of like what you see here. It turns out that there are, within a given stretch of DNA, okay, we can see there's something called 8 histone molecules, that's 8 histone proteins, wrapped with 2 turns of DNA. So each one of these little balls has 8 proteins in it. Those 8 proteins are 2 copies each of 4 identical proteins. So there are 2 copies of a protein known as H1. There's 2 copies of a protein known as H2A. There's 2 copies of a protein known as I'm saying this wrong. Back up. Two copies of a protein known as H2A, 2 of H2B, 2 of H3, and 2 of H4. Okay, so I said H1. That does something different. Okay? That's a total of 8 proteins. Those histone proteins, when they're complexed with DNA, okay, create something we call chromatin. So chromatin equals histones plus DNA. So what you see on the screen is chromatin. Now, these individual things here themselves will coil up. So we get coils of coils of coils of coils. And that's how your cells cram all that DNA in. And when we see a chromosome, what we're looking at are these guys really big complexes together. Very, very big complexes of them together. Chromatin only occurs in eukaryotic cells. Bacteria, which are prokaryotes, do not have that. Now that turns out to be okay for bacteria because bacteria have 1/1000th as much DNA as we do. They don't have the same problems trying to cram all of that DNA into a nucleus, and they leave their DNA fairly bare in the cell. It just lays there. Okay? Eukaryotes have a very different problem because this DNA has got to be replicated. This DNA has got to be transcribed to make RNA. And enzymes have to get in there and do that. And you can imagine an enzyme getting in the middle of this thing and duplicating it, the complexity that would be involved in it. It's very, very complex. All right. We're not going to deal too much with that complexity, so don't worry about that. But when we think about it as a problem, a topological problem, it's a very big problem. Okay. Yes, question. Lisa.
Yeah, I just got a little confused. So the histone balls are what has the protein in it? So
The histone balls are proteins.
of the 8 proteins all.
Mm-hmm. Yep. So the ball has 8 proteins to each of those individual 4. Okay.
Like this. So the video things like this.
It's interesting when we look at those proteins structures are all very similar. And not only are they all very similar, but they're all very similar in going all the way from yeast, which is the simplest eukaryotic cell, to human beings. Okay? If you compare the histone proteins of yeast to humans, you would see there's very, very little difference, which tells us that structure is important for function. That structure has a very important function. We don't want to mess too much with it because if we do, it may not have the right function. It's not going to make those DNAs fit into our cell, and we would have a problem. Okay. Well, what you see on the screen is a plot of the melting temperature of DNA. Okay? We're watching DNA melt. Okay? Well, what's happening here? I take a tube and I shine some light through it, and it turns out DNA will absorb light at about 260 nanometers. You don't need to know that. Okay? Ultraviolet light. It'll absorb some. All right? If I take, and that's a duplex, if I take that duplex and I pull the strands apart, okay, I have the same amount of DNA in there, but now the strands are apart instead of being together, the absorbance will go up. It's called the hyperchromic effect. Hyperchromic. The hyperchromic effect says that when you go from double-stranded to single-stranded, the absorbance goes up. Well, that turns out to be really useful because it tells me that I can detect when DNA is double-stranded or single-stranded by starting to heat it up and watching what happens to the absorbance. And that's what's happening right here. The TM is the it's called the transition temperature. It's also called the melting temperature. Is the midpoint of this transition that you can see here. The temperature at which that occurs would be the melting temperature of the melting point of the DNA. Yes?
So when you say absorbance goes up, you mean wavelength? Or
So absorbance, so when you shine, let's say I take a tube of DNA and I shine light through it. Okay? So absorbance is a measure of how much makes it through. If the solution is absorbing light, less light will make it through. If the solution is not absorbing light, all the light will make it through.
Okay, it's just one wavelength.
So it's just, well, it is done at a specific wavelength, but the absorbance itself is not a wavelength. The absorbance is a measure of how much light makes it through at that wavelength. Make sense? Okay. Okay. So I can tell there where my DNAs get pulled apart. Well, why do I care about that? Well, it turns out to be really useful in some laboratory uses. Okay? This sort of depicts what's happening. Here's a double helix. Here, if I heat it, I pull the strands apart. And here's the kind of cool thing. If I'm very careful, I can actually put the strands back together. I can put the strands back together. Now, the heating and pulling it apart is known as denaturation. The putting it back together is known as renaturation. And why do I care about this? Well, if you think about it, when cells have to replicate DNA, one of the things they have to do is they themselves have to peel apart those strands. We'll see how they do it when I talk about replication a little bit later. But the strands have to peel, the cells have to peel those strands apart themselves. Okay? In the laboratory, if we want to do this, we make it easy. We can peel those strands apart by putting the DNA in some boiling water, and the strands come apart, and then it becomes very easy for us to replicate that DNA. Okay? It makes it very easy for us to replicate that DNA. Now, if you've ever watched the TV crime shows where they get the drop of blood and they get the hair of the crime scene, or they get whatever, and you've heard of DNA fingerprinting, all right? These types of analyses require DNA to be replicated. Okay? So how do I know that it's your DNA that was left at the crime scene? Well, I get that drop of blood that was left at the crime scene, and I take a bit of your blood and I replicate the DNA in both. So I make lots more copies of it. When I replicate it, I can make tremendous amounts more. If I do that, then I can compare the two DNAs and say, well, these two match, you're in trouble. Or perhaps you bled on the crime scene last week and you left it there and we forgot about it, but now you're going to go to jail. So, okay? So that technique in the laboratory where we make lots of copies in the way that I just described is called the polymerase chain reaction. PCR. You probably heard of PCR. How many people have heard of PCR? A few. How many people have done PCR? A few. Okay. So PCR is a laboratory technique that allows me to make lots of copies of a given DNA. It's very easy to do, and part of the process involves this denaturation. So that's an important step in the process of doing PCR. Okay. Let's see. That's what I want to say about DNA. I want to say a few words about RNA, and then we'll turn our attention to replication. Okay, this picture depicts for us some very important cellular processes. And it's got some names on here I want you to not mix up, because I can guarantee you I'm going to ask you about them. All right? Students misuse these names. They need to have some meaning for you. So when I say these names, I want you to immediately think of what they refer to. Well, I've been talking about making lots of copies of DNA, and that phenomenon, I've used the word, is replication. Replication, DNA replication, refers to starting with DNA and making more copies of that same DNA. Cells have to replicate their DNA in order to divide, because each new cell has to have the same DNA that the parent cell had. So if that is to happen, we have to make more copies of it, and that's when we're replicating the DNA. Okay. The next step in the process is called transcription. Now, transcription means starting with the DNA and making a copy of it into RNA. Transcription is not the same as replication. It uses different enzymes, and it has a different product, the product being RNA and not DNA. Okay? One of the reasons that cells are making RNA is so they can make protein. And cells make protein by using the genetic code on one of the RNAs, in this case a messenger RNA, and that process is known as translation. Translation means taking RNA and converting it into protein. That involves the genetic code. We'll talk about those processes in a bit. Okay? But I find that students commonly do not remember what these three processes refer to. Okay? So everybody's clear what transcription refers to, what translation refers to, and what replication refers to. These are very, very important things. Okay? These three processes go together to make something that people refer to as the central dogma. Central dogma. The central dogma says that DNA can make DNA. It says that DNA can make RNA. And it says that RNA can make protein. When I say can make, obviously other things are making it, but they are participating in that process. DNA is necessary to make RNA. RNA is necessary to make protein. Okay. RNA, of course, turns out to be a pretty important player in this overall process because without RNA, we can't make protein. Let's think about the common types of RNA that are out there. Okay? We usually talk about three main types of RNA. Transfer RNA, also called tRNA, is small. It carries on one end a specific amino acid, and on the other end it has a region that makes a base pair with the messenger RNA. We'll talk about that. Transfer RNA, in fact, all three of these RNAs, transfer, ribosomal, and messenger, all three of these RNAs are necessary for making proteins. So transfer RNA is carrying the amino acids to the place where the protein is being made. The second is known as ribosomal RNA. And ribosomal RNA is, there's several sizes. Several of them are long, fairly long. Fairly long meaning a couple of thousand base pairs, or a couple thousand bases, I should say. All right. And as its name would suggest, ribosomal RNA is a component of a cellular structure called a ribosome. A ribosome, of course, is a protein-making complex. It's the ribosome where the protein is actually made. The ribosome has proteins, and it has ribosomal RNA. And they both play very important roles. Okay? The third major type of RNA is known as messenger RNA. Okay? And messenger RNA, also known as mRNA, is what carries the information that gets translated into protein. That is, using the genetic code, the ribosome knows how to convert the sequence of bases in messenger RNA into a sequence of amino acids in a protein. The ribosome helps it to do that, but the messenger RNA is carrying the message, that is the information necessary for making a specific protein. Okay? Now, there are other small RNAs that have important roles in cells. They're known as small nuclear RNAs, small interfering RNAs, microRNAs. And I'm not going to talk about them now. We'll talk about them later in the term. But these guys can play very important roles in controlling whether or not a gene is converted into protein. So they play some control roles. Okay. Well, let's think about messenger RNA here. Okay. This figure shows some very important differences between bacteria and eukaryotes in these processes. Okay? The central dogma occurs in both bacteria and eukaryotes. We make DNA into DNA, we make DNA into RNA, and we make RNA into protein, just like bacteria do. That is, we all have those central components. However, the arrangement of information in bacteria is different than the arrangement of information in your cells and my cells. Okay? I need to introduce a term here. A gene. What's a gene? Nobody's awake today, are they? You're on the right? Yeah? Excellent. Okay. So, a gene is a section of DNA that codes for a certain protein. All right? That's a good definition. A gene is a section of DNA that codes for a certain protein. The differences that we will see between prokaryotes and eukaryotes is how they arrange their genes. Okay? How they arrange their genes. Prokaryotes arrange their genes in clusters. A, B, C. Okay? A is right next to B, is right next to C. Eukaryotes don't arrange their genes in clusters. They arrange their genes usually pretty isolated from each other. That's one big difference. Pretty isolated. So one of the reasons that eukaryotes have chromosomes that are much bigger than prokaryotes is because the spaces between the genes are quite a bit larger in eukaryotes than they are in prokaryotes. Okay? So the spacing is different. In prokaryotes, the genes are not just close together, but they're usually clustered together according to common function. So I'll give you an example. Bacteria have a cluster of genes, all of which are involved in breaking down the sugar known as lactose. Lactose is also known as milk sugar. Okay? There are three enzymes, that the bacteria use to metabolize lactose. And all three of them are located, 1, 2, 3, next to each other. And all three of them are made on the same messenger RNA. This cluster that we see in prokaryotes is known as an operon. O-P-E-R-O-N. We don't see operons in eukaryotic cells. They don't exist. In eukaryotic cells, one messenger RNA means one protein. In prokaryotic cells, one messenger RNA might have coding information for several proteins. It wouldn't be uncommon for one messenger RNA in prokaryotes to code for 10 different proteins. Okay? The last thing I want to say about eukaryotic RNAs, and I'll say more about this later, is that eukaryotic RNAs actually have the coding information frequently in pieces. You'll notice up here, gene A is one piece. If we look down here at gene A, one piece is here, one piece is here, and there's something in the middle of it that doesn't make it down into the final messenger RNA. It gets cut out. It gets what we call spliced. So what's happening is, here's that DNA, here's that RNA that's made from it, and then that RNA gets a section chopped out of it. It gets spliced out, and the final messenger RNA doesn't have that sequence. Now, we'll talk about that splicing process later, how it occurs. But a big difference between prokaryotes and eukaryotes is splicing. Prokaryotes don't splice. Eukaryotes do splice. Prokaryotes have all their genes intact, and eukaryotes have all their genes in pieces. Yes, ma'am? So an operon is an arrangement of genes that we see in prokaryotes where the genes are clustered together on one messenger RNA. Okay, I'm going kind of fast here. Why don't I slow down? Yeah. Give you a chance to catch your breath. Your hands hurting? Questions about that? Is there a question back there? No. What are you doing this weekend? Going where?
Just maybe enjoying the sun.
Enjoying the sun. There's wishful thinking. What's that? Nobody's doing anything fun this weekend.
Farmers market.
Farmers market's nice, yeah. If it's not wet. Planting what? Starts, yeah. I need to get my tomatoes out. Extra credit? Did I hear extra credit? You want extra credit this weekend?
Yes, please.
I can do a pop quiz, I suppose. Well, I mean, if you get any credit then on the pop quiz, then that's extra, right? That was nervous laughter, it sounded like. I'm open to that. How about that?
Okay.
Take out a blank sheet of paper.
Okay.
And on that blank sheet of paper, I want you to write your name. Okay, write your name. And question number 1, I want you to write your name cursively backwards. Fun exercise.
Like just the letters backwards, or you want to like mirror backwards?
I think writing the letters backwards, yeah. It's kind of fun. And I'll check for spelling, so you got to spell it right too.
Are we writing the letters backwards and the name backwards?
The letters are in your name, right? Oh, yeah, no. So you can write the letters in the right normal way, but I want you to. What do you? I'll tell you what a fun exercise. Seriously, try doing this. Try writing your name upside down. Okay. Somebody tell me about this. That apparently a lot of doctors and hospitals train themselves to write their names upside down. So when somebody gives them something to write, they don't have to turn over. They just write their name upside down, and they've got it. And I thought, you're kidding me, right? And so I tried it. And within about 2 minutes, you can train yourself to write your name upside down. I'm not kidding. Try it. It's really weird. And before you know it, you can write your name upside down. I'm not asking you to do that, okay? But I'm writing, just write it in reverse order, the letters of your name. Just in cursive writing, all right? So the last letter should be a capital, right? Like my last name would be Nriha Nivek. I'm not going to grade it too closely. Okay. Second question. How many points should this be worth? It's got to be the right answer. Uh-oh. Now you think, oh, my God. What's the right answer? So whatever I pick is the right answer. You'll get points if you got it. Ooh, wouldn't that be weird? If you write down the number of points, what number between 1 and 10 am I thinking of right now? Right? So I want your opinion. How many points should that be worth? Reasonable answers will get credit. Whoa. And I think that'll do it. How's that? Okay. All right, so I'll get those at the end of the period. Yeah.
What's the benefit to us splicing
Yeah.
genes over them, just putting them all together?
It's actually a very long answer. I won't give you here. There is a benefit, but it'll take me longer than a minute to do it. Come talk to me. I'd be happy to tell you, though. Okay? All right. I hope he wasn't leaving for the day. What's that? Oh, okay. All right. See, that would be bad form. That would definitely get you some minus points. Okay. Now, second RNA that's important, transfer RNA. Transfer RNA has a sort of a schematic structure that we describe as a clover leaf. Okay, a clover leaf. Look at that. All right. Notice that this RNA is one strand. There's the 5' end. We trace it all the way around. And there is the 3' end. What are those bars in the middle? Those are hydrogen bonds. Those are base pairs. So we see that, excuse me, we see that a single-stranded nucleic acid can form base pairs with itself if the base pairing information is there. So it's possible for that to happen. And so transfer RNAs actually have things set up where they can form base pairs inside themselves, and it gives the transfer RNA a characteristic structure like what you see there. Looking at this, there are two main features of this structure. One structure is the 3' end, and it's at the 3' end where the amino acid is attached. The other part of the structure that's important is down here in section 2. Okay? In section 2, this region is called the anticodon. The anticodon. Well, I haven't told you what a codon is, so I better tell you that before I tell you what an anticodon is. The genetic code is written in units of 3 nucleotides. A, U, G in an RNA is a codon. So that 3 nucleotide unit is a codon, and that 3 codon unit is complementary to a 3 codon unit in the anticodon of this transfer RNA. They have to form the proper base pairs. A pairs with U, U pairs with A, C pairs with G. Excuse me. Okay? Now, during the process of translation, this guy has got to pair properly with the right sequence in the messenger RNA. If it doesn't, it's not the right transfer RNA. So it turns out that the anticodon that's down here is linked specifically with a specific amino acid up here. That's how the genetic code is made possible. A specific amino acid goes with a specific anticodon. That means that different transfer RNAs will have different anticodons. One that has a glycine up here is going to have a different anticodon than one that has a tryptophan up here. Okay? That's what makes the genetic code possible. Okay. There are modified bases that occur in transfer RNAs. Modified meaning chemically modified. Okay? I'm not going to make you know those. Don't worry. The last of the major classes of RNA I want to talk about are the ribosomal RNAs. Ribosomal RNAs are also called rRNAs. And you look at this guy and holy moly, that guy is complicated. This is one strand. And look at the base pairing that happens inside of that strand. There's one end of it, there's the other end of it there. Okay? Ribosomal RNAs are really wild. They're not random. It's kind of like we talk about proteins. They're not random. Yes, sir?
So you just said it's not random, so it does that every time?
It does that every time.
That exact thing?
That exact thing. Holy crap. I think you express it beautifully.
It's like the building plan for like a fruit maze or something.
It does. It looks like something out of outer space, right? Now, what did we learn about structure of proteins? Essential for function, right? We disturb structure, we lose function. The fact that this makes this same structure every time is kind of magical, but it's no more magical than a protein making the same structure every time, right? It also tells us that if it makes the same structure all the time, that structure must be important for function, and it turns out, yes, it is. Okay? Now, I'm not going to tell you what the functions are here. There's 2 big functions that we'll talk about with ribosomal RNAs. I'm not going to talk about those here because we'll talk about those during translation. But needless to say, holy crap, that is an odd structure, and yes, that structure is very, very important. And if we look at that structure in a bacterial cell and we compare it to a structure in a human cell, it's not very different. Not very different. Okay. I'm going to come back. I'm not going to talk about snRNAs and snRNPs at this time. I'll talk about RNA interference later. Okay? Okay. That covers what the general things I want to say about RNAs and DNAs. I'd like to turn our attention to talking about DNA replication, but I thought we might first do a song. Okay? So I have a song. It's to the tune of an old Simon and Garfunkel. Oop, wrong thing. All right? Called Feelin' Groovy. I hope you guys know the tune. The DNA forms A and B. They have bases complementary. Despite the similarities, they differ in their major groovies. Na 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 na. Major groovies. Transcription factors with their binding cause DNA to start unwinding, holding it aggressively. by forming bonds in major groovies. Na 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 na. Major groovies. For proteins, the key to sequence ID is hydrogen bonding, each base pair unique. Purine, pyrimidine, patterns discrete. In DNA's most major groovy. Na 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 na. Major groovy. What I didn't tell you in the structure of DNA, of those DNAs, was that when the double helix comes together, it makes a big, there's two grooves that form, something called a major groove and a minor groove. The major groove is a big gap. The minor groove is a little tiny gap. And you can actually see the difference between them. And cells, the proteins of cells, use those grooves as ways of reading the sequence. It's pretty amazing what they do. Okay. Dude, what am I doing? My brain is not working today, guys. There we go. Replication. So we've got about 10 minutes. We can get started on this process, and we'll finish it up next time. So we talked about 3 steps in the central dogma. DNA making DNA. That's what we're going to talk about here. DNA replication. DNA replication occurs in a fairly orchestrated process inside of cells. There's our central dogma again. It's shown in another way. DNA makes DNA. DNA makes RNA. RNA makes protein. There's some added things on here that screw us up. Okay? The central dogma, when people first discovered it, said, oh, we know the secrets to all there is to know about nucleic acids. And then in the 1970s, they discovered something that really surprised them. There were some violations of the central dogma, meaning that it wasn't one-directional. It didn't just go in one direction. But in some cases, it goes in the other direction as well. Okay? That's what you see right here with this little purple line. Reverse transcription. What does that mean? Well, if transcription means DNA making RNA, then reverse transcription must mean RNA making DNA. And it turns out that does happen sometimes. It happens in something that you don't want it to happen in. Okay? It happens in something that you don't want to have it happen in, and that is retroviruses. There are certain viruses that have their genetic information in the form of RNA. And during their replication cycle, they get converted from RNA into DNA by an enzyme that they carry that's called reverse transcriptase. Transcriptase is spelled Transcriptase. Reverse transcriptase is capable of using RNA as a template, that is something it copies, and making DNA from it. Okay? Makes RNA as a template and copies DNA from it. All right? Now, that reverse transcription is very important for those viruses. Examples of retroviruses include HIV. HIV is a retrovirus that has an RNA genome, and part of its replication cycle, it goes through a DNA phase. Some of the strategies for defeating HIV are to inhibit the reverse transcriptase from functioning. So some of those strategies, it's a viral enzyme, it's not a human enzyme. If we can knock that enzyme out, we can stop the virus from replicating. Another violation was right here. RNA replication. Again, when we look at viruses, we see some unusual schemes. Some viruses that have RNA as their genome, that is, as their genetic information, don't go through a DNA phase. They simply copy RNA and they stay completely in the RNA realm. Okay? So those 2 violations of the central dogma are known. There's no violation that turns protein into RNA that anybody's aware of. And there probably won't be, because that's a very different kind of a process. Okay? So that's the central dogma as it exists today. I'd like you to think about how replication occurs. This is kind of a nice diagram. Right? Here's a bacterial DNA. Bacterial DNAs are circular. They're circular. So when we look at the DNA in an E. coli cell, it's circular. Replication starts at a specific place. There's a specific location. It has a specific sequence of bases. And that specific location where replication occurs in any DNA is called the origin. Oh, well, it's spelled on there, O-R-I-G-I-N. When we look at how this occurs in bacteria, it's what we call bidirectional. It starts here, okay? And then we see one replication fork moving to the right and down, and one moving to the left and down. Okay? So it's bidirectional. And what will happen is these guys will come around here, and they'll finish down here, and when they're done, you'll have two circles of DNA. Okay? Eukaryotic cells have a very different kind of thing. They do also do bidirectional. You see, they start here, here's an origin, and it spreads out. But instead of having one origin, like bacteria have, our cells have hundreds or thousands of origins because we have much longer chromosomes. Okay? So that's a very big difference between us. We both do it bidirectionally, but we have many origins. Another big difference, and we'll talk about it later, is the fact that our chromosomes are not circular. They are linear. They have ends. And that turns out to be something that's very, very, very important when we think about human health, we think about aging, and we think about cancer. And I'll talk about why linear DNAs are really a big consideration when we think about how DNA replication occurs. The enzymes that make DNA, that copy the strands of DNA, are called DNA polymerases. P-O-L-Y-M-E-R-A-S-E. A DNA polymerase knows when it encounters in the strand that it's reading an A, it knows that a T's got to go next to it. When it reads a C, it knows it's got to put a G in next to it. So DNA polymerases are doing several things. One is they're reading what's called the template strand. That is the strand that they're copying. They actually read it. Those hydrogen bonds I was talking about before tell it what base it is seeing. Okay? The second thing that the DNA polymerase is doing is that it's making a phosphodiester bond like we talked about before. It's going to join the hydroxyl to this phosphate and join these two guys together. So it's catalyzing the formation of a phosphodiester bond. Now, amazingly enough, most DNA polymerases also, in addition to reading it and making this bond, proofread what they do. They proofread what they do, meaning that if they make a mistake, they will stop, they will back up, and they'll remove their error, and then they'll go forwards again. Now, what's remarkable, okay, that's all cool. 3 cool functions, right? What's remarkable is, in E. coli, they can do that at the rate of 1,000 bases per second. 1,000 bases per second. Okay? They're doing it at the rate of 1,000 bases per second, and they make errors about 1 in every 10 million. Imagine typing 1,000 characters per second, which you couldn't begin to do, and making 1 error every 10 million keystrokes you had. You would be a very popular typist if you could do that, and you would be a very accurate typist. Okay? So DNA polymerase is doing some pretty cool things in that respect. It's been a long day. It's been a long week. I say we cut it short. If you did not get your exams the other day, follow me back to my office. I will give them to you.